Good morning, everybody. Today we have Pablo del Pino. Pablo, he got a degree in physics at the University of Sevilla in 2002, and then he did a PhD in molecular biophysics in the Technische Universität München in 2007, supervised by Professor Fritz Parak. Then he did a couple of postdoctoral stays, one with Professor Wolfgang Parak in the Center of Nanoscience in München, and then in AG Biophotonic in Philipp Universität Marburg. Then he did another postdoctoral stay with Jesús de la Fuente, Instituto de Nanociencia de Aragón. And then he became an ARAID researcher in 2013, then co-leader of the bioengineered particles laboratory in CiQUS biomaGUNE. And then he became Ramón y Cajal in 2013, and 15. And he has been working in Condensed Matter Physics Department and CiQUS. And then he promoted to associate professor in 2021, and currently he's a professor at CiQUS, a Center for Research in Biological Chemistry and Molecular Materials. And his research interests are design and development of nano and micromaterials for biological and medical applications, materials for therapy, imaging, and biosensing, and in smart multifunctional materials at the interface of material science, biology, and medicine. Today he's going to talk about multifunctional MOF-based nanodevices. It's a pleasure to have you here, and looking forward to your talk.
Thank you very much, Pepe, for the kind introduction. It's a pleasure to be here. Thank you very much for attending my talk. So together with Beatriz Pelaz, that is there. So this is Bea. We have a group in CiQUS. This is the group, the people that has done the things that I'm going to present today. Yeah, the group is called BioNanoTools. So what we do is nano things for bio things. Basically, we make nanoparticles. This is CiQUS, in case you know. This is a building in Santiago de Compostela. It's a very nice institute with a lot of people that work in organic chemistry, but then we're working in material science. And then us and one more group working with nanoparticles. These are the two labs that we have. In this lab, we do materials. And in this one, we test them in biological systems, in 3D bioprinted tissues, cell cultures, and things like that. So now at this point, we created the lab in late 2016. And Bea and me, we come from the inorganic nanoparticles area. So basically, we're making libraries of inorganic nanoparticles, metal nanoparticles, quantum dots, magnetic nanoparticles, and so on. And then we investigated how small variations in these nanoparticles affect internalization, trafficking, toxicity, and these sort of things. So we basically work before coming to CiQUS in organic nanoparticles and functionalization of nanoparticles, and then with polymeric capsules, where we could also put nanoparticles to make them smarter. But then we came to CiQUS, and we decided to open new lines. And one of the lines that we opened was about porous materials or porous nanocrystals to make applications in biological applications. This is my most personal research line in the group. Then Beatriz works with DNA nanotechnology, making origamis and motors, molecular motors, and these sort of things. And then we have another one that is about biomimetic nanomaterials. We make nanoparticles with the cell membrane. Okay, but today I will focus on MOF-based nanoparticles. So this is an outline of my talk. So I was asked to give a brief introduction of 15 minutes, very general about the topic of my research. So this is what I will do at the beginning. Sorry, because I forgot to put the timer, and otherwise I will be talking a lot. And then I will be talking about stimuli responsive MOF. I already told you that we come from the background of making nanoparticles that you can excite with light, magnetic fields, ROS, and these sort of things. So I will be talking about MOF for these kind of things. Then briefly, one example of how we can use this kind of core shell inorganic MOF systems to make engaging chemistry, so to activate prodrugs. And then finally, I will talk about recent papers on how to functionalize nanomorph to make them go to or follow specific internalization routes to make them more efficient to internalize in cells and these sort of things. Yeah. So the motivation of my work has been for almost 20 years is to make nano things that can interact with light, magnetic fields, and other kind of external stimuli, and also internal stimuli. The idea is to make smart materials. One of the issues that we always had has been that when you feed nanoparticles to cells, they always go to the same place. They go to a lysosome, that is a very acidic organelle where you have a lot of enzymes, the degradative enzymes, and a very basic pH. So when we were working with inorganic nanoparticles, normally this is not an issue, but we have to know that they go there. So they don't have access to the rest of the cellular machinery to do things. So probably you know with the COVID vaccines, they have to engineer a lipid nanoparticles to be smarter enough to not follow strictly this route so that the messenger RNA can escape from the lipid nanoparticle and do what the messenger RNA is supposed to do. So, but already back in 2007, we were designing this kind of polymeric systems where we have plasmonic nanomaterials. They were kind of nanotriangles of gold that absorb a lot of near-infrared light. And then we were putting these near-infrared nanoparticles on a polymeric shell. And inside we could put polymers, fluorescent polymers, drugs, and these sort of things. And the idea already almost 20 years ago was you have here 4 cells, and then in this one, you can see that you have a black thing that is a polymeric capsule with gold. And the idea was to go with a pointer, a light pointer, go through the capsule and open it and release the thing so that the polymer can go outside, and then you have access to the whole cytosol of the cells to do whatever you want to do, gene therapy or whatever. If you don't put enough energy or irradiance there, you pass with a light pointer through the capsule and nothing happens because you have to put enough energy there to destroy that. Now I change to the fluorescence channel. You see the laser now. This is this spot that you see, and then you see something comes out from the capsule. And this is what happens before you use the light pointer and after you use the light pointer, okay? So this is the first thing that I did as a postdoc, that was to build a microscope with a laser, just to be able to do that and pass with a light pointer kind of similar to this one to do that. Well, these kind of capsules, these smart capsules, are very interesting, but they are micrometer range. We didn't have a lot of access. And then when we came to CiQUS, actually 1 year before we came to CiQUS, we started collaborating with Patricia Orcajada, that is one of the pioneers on BioMOF, or MOF for biological applications. And then we started to think about developing similar systems with nanoparticles and MOF. At that time, I'm telling 10 years ago, there was not a lot of literature of studies making libraries of nanomorph for biological applications, not at least as it was with inorganic nanoparticles. And we started collaborating in trying to make nanomorph that has a lot of interesting properties for bio applications. So we started this something like 10 years ago. When you downsize MOF, new, interesting, and new to discover properties appear. Very interesting, not like with quantum dots, but very, very interesting. It's very interesting also that you can control the defects. The surfaces are very reactive. They are not completely full, the places. Then you can also do surface functionalization as we were doing with inorganic nanoparticles. Okay? Imagine, like in the case of gold nanoparticles, you use sulfur gold chemistry. You can do a lot of things with gold nanoparticles. With magnetic nanoparticles, you can do a lot of different things. With nanomorph, you can do also a lot of functionalization. So we are working, I will show you what we do now. But already since the beginning, what we wanted to make is core shell nanosystems. So my idea from the beginning was to have one of my very cool nanoparticles, plasmonic nanoparticles, magnetics, and so on, and then to grow a MOF around it. So the idea was to develop a seeded growth approach. Like when you want in a surface to grow something with control, but I wanted to do it on top of the nanoparticles. So at that time, we thought of a lot of, because I was discussing with Nuria before, of modifying nanoparticles just to grow in a layer-by-layer mode or in a controlled way. Nothing worked. Nothing worked. And eventually, we end up working with CIF8. I will come back to this. So basically, with nanocomposites, this is what we started. We have two approaches. You can make the nanomorph and then put metals inside. But for me, this is not interesting because the typical microporosity of MOF is too small to have inorganic nanoparticles where you have enough electronic state or sufficiently dense to have nanoproperties, okay? Because typically you need more than 2 nanometers to see interesting gold properties or magnetic properties and so on. So this is not something interesting for us. And then we decided to go to the core shell approach, that is what I was mentioning before. So it was not trivial at all to do this, because what we found is that the common or most commonly used synthetic route to make MOF are not compatible with the stability of metal nanoparticles. So basically, you have imagine, you have a gold nanorod, you make the MOF, and if you find the cool conditions, you will make the core shell thing, but you will end up with a sphere coated because you completely deform the conditions. And this is because normally we use DMF, 120 degrees, a lot of time, pressure, acids, and things like that. So nothing worked, but only worked with this MOF, this zeolitic imidazole framework, that is very easy to do. And there are thousands of papers with this MOF, thousands. Everybody uses it for drug delivery, for a lot of different things. It's very easy to do. So basically, you have to mix with CTAP, a surfactant, and you will have beautiful cubes of CIF8. And because we know that for 30, 40 years, we use CTAP for growing nanoparticles, if we use CTAP-coated nanoparticles, we can grow CIF8 on top of it. Okay? Any nanoparticle, as long as we put CTAP on top of it, we can grow the CIF8. There are papers in JAX and so on explaining why this happens with the micelle concentration of CTAP and the kind of structures that you do, but you end up with beautiful nanoparticles with this. So this is the kind of first study that we do. So we make a gold nanostar, and then we grow the CIF8 on top of it. This beautiful image is a courtesy of Sarah Valls in Antwerp. So you have a gold nanostar, and then you see the structure of the CIF8. In this first study, what we wanted to try is if we were going to be able to do what I show you with the microcapsule, so to put a light pointer on the system. In my opinion, by thermodiffusion, we're going to be able to expel, to release what we had encapsulated inside the MOF. Okay, MOF, this case, C8 has small pores of about 0.5 nanometer, 1 nanometer, and you can encapsulate a lot of different molecules. The kind of molecule that we wanted to encapsulate, in this particular case, is a HOH. If you are familiar with biology or whatever, this is what we use to stain the nuclei of cells. Basically, it has a very weak fluorescence, but when it coordinates with DNA, fluorescence go up like crazy. So for us, initially, it was a reporter to know if we had leakage of the dye. So imagine you have the MOF, you put it to the cells, and then suddenly you have a lot of fluorescence in the dye. It's because the dye went out like crazy. That is what happened. Okay. So I was very naive at that point, and it happened that all the papers is not true. So basically, C8, as long as you put it in water, it is destroyed. It is destroyed. So it takes seconds to be destroyed. I will show you a little bit why this is destroyed. Because the synthesis is made in water. So you made it in water. You clean it with ethanol. You dry it, and then you put it in water, and it's destroyed in a matter of seconds. It doesn't have sense. So basically, we had to come with an idea to sort this out, and we use amphiphilic polymer that we have been using for 20 years. It's a PMA. We call it PMA. It has an hydrophobic change and hydrophilic tails. And basically, we coated the MOF, and then with this coating, we prevented for weeks, for months, the stability of the MOF. But we had to put a polymer coating on top of a MOF. That is not something that I wanted to do. We did it, and then it is stable in very acidic conditions, in media, mimicking the kind of environment that you have in the lysosomes and these things. In water, in cell medium, and so on. Here you have the colloidal stability up to 1 week of the particles, and with this polymer coating, they are stable. So now, what I want is to put some light. This is the kind of system that we use. This is a system where we can control very well the spot size to illuminate few cells or thousands of cells. And this is what you have. In orange, you see the membrane of the cells, and then you can see that you have some cyan dots. These are the nanoparticles. Okay, this is the MOF, where you have the weak fluorescence of the cargo that we put on top of inside of the MOF. Then I put some light, and I could release the dye from the cell. So my idea was working there. It can be, this is a very long paper. By thermodiffusion, it might be other explanations to explain why the drug that I had encapsulated can go outside of the particle. My idea is that I have something that is very hot in the middle, and I create a thermal gradient that is able to release what you have encapsulated in a kind of spongy structure on the nanoparticle. In collaboration with the people in Toulouse, we have also done this with magnetic nanoparticles. Again, it's the same system. We make nanoparticles. It can be cubes, spheres, rods, tetrapods, whatever. As long as you put CTAP on top of it, you can grow C8. This is the same with different detectors. And you see that in all of these kind of cubes, you have a magnetic core in the middle. Okay, this is what we wanted to do. And what we demonstrated also here is that the particle is the seed. By controlling the number of seeds, you control the final concentration of particles. If you put a lot of these seeds, you will end up with a lot of nanoparticles, because in each seed, you grow the MOF. If you put few of them, you will make a thicker shell. Okay? So we have demonstrated also with alternating magnetic fields and rotating magnetic fields. So and we are also able to destroy tumors and release a Prof. And I wanted to go to this one because it's again the same system. So we do it with light and with hyperthermia. Then in a collaboration with José Luis Mascareñas, we wanted to test if we were going to be able to do very complicated reactions inside a living cell. So José Luis, for those of you that don't know José Luis works in chemical biology, and he's an organic chemist, and he makes molecules and try to do chemistry inside living cells. And then he prepared some molecules, 1 and 3, so that typically you can close them and make them fluorescent. But this kind of reaction, this thermocyclization, requires 90 degrees organic solvent overnight. So my challenge was, can you do that inside a living cell? And my idea is that, okay, I have something that is going to be very hot, a golden star. I will put some light, and I will create a very high temperature gradient where I'm able to do these reactions. So in a tube, it works very well. So we produce the product in 100% quantitative yield. So I'm able to do in a solution with my nanoreactors, put some infrared light and convert the product. So I can make 2 and 2 and 4. So these reactions happen because I put a lot of temperature. But I'm doing this in a solution where I don't increase the temperature a lot. Okay? So I want to make confined nanoreactors that are activated by light. So I put light, I heat the core of the nanoreactor, and then the molecules that are close to this nanoreactor, that in this particular case I have a MOF that takes a lot of these organic molecules because C8 is very hydrophobic. So if you put something hydrophobic, it will go inside. And then I can generate this product. And then we try to do that inside the living cell, and this also worked. Okay? So we were able to make this kind of reactions, but inside the living cell. And this is the ultimate proof that I'm not increasing the temperature a lot. Because if I put a cell for few minutes above 45 degrees, 44 degrees, I will kill it. And remember that I need 90 degrees overnight organic solvent to produce these things, and we are able to do that. These are the products. The green are the products. We were able to do that with near-infrared light. Okay? So yeah, these are the kind of reactions that I told you that we do to produce 2 and 2 and 4. And these are the different controls that we use to prove that it's only because of the combination of the nanoreactors with the light that we can do these uncaging reactions. Okay, so already more than 20 years ago, no, 20 years ago, 12 years ago, it is known that C8 is not stable in water. And the reason because it's not stable in water is because of the CO2. If you have CO2 in the water, CO2 chelates zinc and destroys it. I didn't know that at that time, but when you put a polymer or you put something that prevents CO2 absorption or takes CO2, you prevent that you destroy the MOF. So any zinc-based MOF with CO2, we have to be careful, and that's the reason because we use this PMA coating that allows us to do a lot of work in that direction. Now we are working in improving this, because something that I was astonished is that if you put a solution of your C8 as you synthesize it and you put it in the incubator of cells, when you remove it, what you have is like a MOF Gruyere MOF. You have a lot of holes inside. We discovered that because in Santiago, you know that it's very humid, and sometimes we forget some samples on the table, and then when we want to see it, suddenly we start seeing cubes with a lot of holes inside. What is this? And was because of the humidity that you have, the CO2 that you have in the atmosphere. If you leave it there, eventually it will degrade. Of course, if you put it in water and you don't remove the CO2, or you put it in an incubator with cells where you have 5% of CO2, because cells have to be cultured with 5% CO2, it's a matter of minutes or hours that you destroy the MOF. And I want to put this on a paper because it's very interesting. Here you see original C8. These are relatively large nanoparticles because I wanted to see the holes. And if you put them in contact with a lot of CO2, you end up with these capsules. It's completely destroyed. If you leave it inside the incubator, from this, you go to this. Can you see the holes? And this is what happens, is that we have a new zinc-based structure. I have found a similar diffractogram, but we are still working on that. So you go from this simulation when you synthesize it, and suddenly when you put it in the CO2, you start you have a new phase that is not porous. And that's the reason, because this is BET, the absorption of nitrogen of the original MOF. After 1 day, 3 days, and 7 days in the incubator, you decrease completely because you are getting a new material. It's something that I want to publish because the people still are publishing drug release with C8. A lot of papers every day. And it doesn't have sense. If something that you put in the cell culture immediately is like sugar, you can actually put the drug directly. It doesn't have any sense. Okay, but then Manuel Ceballos is a postdoc now in my group, but he did the thesis. He came and he had the challenge to make the core shell systems, and he has actually succeeded. I will briefly talk about this zirconium MOF. So basically, Manuel has been working in, he can make nanoparticles, B pyramids, rods, stars, any gold nanoparticle or magnetic nanoparticle. But the idea was to work on the surface to try to have some kind of interaction either by the linker of the metal cluster of the MOF to promote the crystallization of the MOF on top of the nanoparticle. And he has tried a lot of different things. Everything that is in the literature, a lot of different things. What we found is that, I already mentioned that, is that if you increase the temperature, that is normally that you need something that you need for making this zirconium-based MOF, you change the morphology of the particles. And then they are not interesting for me. If I have a rod or a B pyramid or something that has very cool properties in the near infrared that I need for excitation, if I end up with a sphere that has the absorption in the green, it's not useful for me, okay? Because I don't want the spheres. I want, in the case of magnetic nanoparticles, it's fine. But if I destroy the core when I'm doing the nanoparticles, it's not useful for me. So he found, and at the beginning I didn't believe it, that by pegylating the nanoparticles with PEG, that is something that we always do, these nanoparticles can sustain increased temperatures. And then he has developed different methods, either by preparing the cluster, the zirconium cluster before, trying to minimize very fast reactions of not 2 days, but half an hour, and so on. He has been able to grow this MOF. This is the porphyrynic MOF on top of any nanoparticle. And then he has been able to grow rods, cubes, magnetic nanoparticles, quantum dots, anything. For instance, with this pyrene-based MOF. This is NU1000, is a very famous one. And so last year, we published a couple of papers on this, and I will briefly tell you what we did. What is very cool about this MOF is that they are very, 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 very stable. Zirconium-based MOF, they sustain very acidic conditions, high temperature. There are thousands of different MOF based on zirconium-4. These two particular ones, PCN224, no, but PCN222 and NU1000, they are mesoporous, so you have now big cavities where you can even put small proteins of 3 nanometers. It's not the C8 that you have very small cavities. And with this PCN, you have also the porphyrins that are very cool for photodynamic and for many of the kind of applications we do. So Yeah, so the first one that he worked is with this PCN224. That is a cis-connected MOF, has very large porosity. You see here the structures you have. And these are the kind of structures. So the first thing that we wanted to prove is the seeded growth approach that I told you. So basically you have one B pyramid, one nanoparticle after the synthesis. And okay, maybe there is one where you don't see the B pyramid, but more or less for each nanoparticle you can have the core-shell system. And he demonstrated that when he doesn't put the nanoparticles, you have the system. When you put few nanoparticles, this is one, is the picomolar, it's something internal, 1, 5, or 10. It's exactly the same synthesis. The only thing that he changed is the amount of seeds that he put. He changed the size of the system. Okay, so he can control synthetically the growth or the thickness of the shell of this particular MOF. And as we did before, and you can imagine for this MOF, we have a B pyramid that has a lot of interesting properties in the near-infrared light, and we have the porphyrin. So we could use two excitation sources, a green laser, red laser also works, and a near-infrared. The green one for exciting the solid band of the porphyrin to create ROS and kill the cells with a green laser or red laser. This is not very interesting, but you see here what happens. These are, if I'm not mistaken, this is a scan of 100 images of the cell. Okay, you see a lot of squares, and then we only excite the porphyrin here, and when we excite it, we kill the cells. Okay, by generating reactive oxygen species of the porphyrin excited in the solid band. But then we can also encapsulate a drug. And then you will see, we have an scanner. Now we have something more advanced. And basically you tell to the microscope to excite with an infrared light one cell or two cells or whatever like that. Or thousands of cells in a very big window. But he does it automatically with a scanner. He excited this region of interest, 1 and 2. And this is before and after, before and after. So again, with this MOF, we were able to do exactly this drug release of the system. Then with this PDN-based MOF, with this NU1000, where you can encapsulate a lot of different things. Manuela is another PhD student that she finished the last year. She wanted to go a little bit further in the biological testing. And I asked her to compare the CIF8 system with the NU system. So basically, they prepare, this is the CIF8 with a B pyramid and the polymer coating. And this is the NU without the polymer because you don't need it, and the B pyramid. So they are equivalent system, kind of similar on size, and we wanted to compare it. Okay, so it ended up that the NU works better in terms of the dose that you can put to the cells and these sort of things. And again, we do the same. You see, this is before excitation, and this is after excitation. So with the scanner or with a laser, we can go and excite and produce, release the content of the MOF inside the living cell. I said, okay, now I don't want a fluorophore. I want doxo, carboplatin, oxaliplatin, and whatever. And I ask it now to tune the radiation dose to not kill the cell. Because you have to be very careful. If you hit a lot with a laser, you will produce a lot of heat and you destroy. This is a hyperthermia, something that you want to do with magnetic fields and so on. So I ask it, okay, you have to tune the radiation, the time, the dose, and so on, so that you have this scenario. Okay, this is the control. It's the particle without anything inside. So nothing happens. You look at 24 hours, 48 hours, and you see that with this irradiation, very low irradiation, you don't kill the cell because you don't produce enough heat to kill the cell. Because what I want is that what produces the cell death is the release of something that is a drug that is used in the clinic, doxorubicin or carboplatin or This is 1 minute, 2 minute radiation and so on with a very controlled light dose. And she was able to only kill because of the release of the drug. It's the only explanation possible. Okay. And then I asked her, okay, now try to do this in a spheroid. If you don't know what a spheroid is basically you grow the cells and you don't let them spread and they form a ball of cells. By putting different kinds of cells, you can recapitulate a little bit the status of a solid tumor. Okay, where you have a very POSIC region of the core, and then you have different things. It's a model more advanced, or the next model after doing 2D cells to study drug delivery system and things like that. So she did that, and now the idea was to destroy the tumor, the spheroid, only when you have the system with the drug. Not because of heat, but exactly what I told you before with 2D cells, adherent cells, but in a tumor. So this is what happened before irradiation. You have this tumors, spheroid, and after 72 hours, they look like that. And then we have this other one, is the same one as this one, where you have, after 72 hours, they look a little bit smaller or whatever. After irradiation, we want to destroy the tumor. And this is something that he achieved and demonstrated by looking at the spheroid diameter, how big it is, and then to measure the amount of live cells that remain after the treatment and the release of the carboplatin in this particular case. Okay, so he demonstrated that with this MOF, they can penetrate inside a model of a solid tumor, not because of heat, because if you don't put the drug, you don't destroy the tumor. You need the light and the drug. So I have finished now with light and external fields, and I will show you one example of engaging chemistry, and it's based on palladium. The group of Jose Luis is specialized on palladium chemistry and use palladium as a catalyst for making reactions inside living cells, for building molecules, activating molecules, and so on. In case you are not familiar, engaging chemistry is a term that has 10 years, more or less. And basically, you have a molecule that is not toxic, it does nothing, it's not fluorescent or whatever, that then you do some chemistry, remove one capping group, a propalkylic group, for instance, you remove it by palladium chemistry, and then you get a molecule that is toxic. Or a drug that is very commonly used in the clinic. You can destroy the functionality by capping one of the reactive groups and have a bond that you can be destroyed by palladium chemistry, by light, by whatever. Okay? So the idea was now, again, with collaboration with Jose Luis, he has this molecule where you have these molecules that are fluorescent, but when you put them a propalkylic group, they become not fluorescent. Okay? And the idea was to have a palladium catalyst, heterogeneous, that was most advanced than what they use in the lab to produce 2 and 6 that are simply fluorescent products. But these things, you can do it also with molecules to become active or toxic or whatever. Okay? In our particular case, it's also true that this is toxic. So we have these palladium cubes that are very nice in shape and so on, very easy to do and so on. Then you do the reaction, and you do one reaction to produce these things, and they are completely destroyed. Okay? The cubes, simply by being catalytic, they get destroyed after the reaction. Okay? It's a lot of controversy with palladium catalysts and what happens with them, particularly when they are nanoparticles. So our idea was to use the method that I told you to grow the MOF on top of the palladium cube to protect the nanoparticle and have like a nice sponge where the starting material can go in or has affinity to go in, get in contact with the surface of the palladium, do the catalysis of palladium, and go outside. And do this inside the living cell. Okay? So this is what we did here. In the case of the MOF, here we have in the middle a nanocube of palladium. This is before the reaction. You see that you have little blue, and after the reaction, you have this fluorescent product. And the same with this other reaction. Okay? So Jose Luis, the first thing that he told us to do, okay, you have to do this in cell lysates, in PBS, and so on, because this kind of reaction works very well in organic solvents, and we wanted to do in aqueous solvent with cells. So actually it worked very well with the two reactions, and this is the concept where you have a cell where you insert the palladium nanoreactors, that is a nanocube with a MOF on top of it. You have it inside a living cell, and then you start adding the substrates and see if you can produce inside the cells the fluorescent product. This is what we did. We produce the green product, and we produce also the pink product. These are the two kind of reactions that we do. It works very well, and the more time that you left the substrates, the more fluorescence that you get. We compare it with palladium nanoparticles, and it doesn't work. You see, this is the nanoreactor, this is the palladium nanoparticle, and these are three widely used homogeneous palladium catalysts. Okay, just that you see, we put the same amount of palladium and see how much of this we could get. And something very cool is that with the same cells, we could do one run to express one fluorescent product, clean it, and make a second one. And the cool thing about this is that we could do these reactions for a week. So basically, you have a cell culture with your palladium catalyst inside, and then you start adding substrate, checking the microscope, clean, and then when you clean, by in a concentration-dependent manner, because different chemical gradients, what you have inside the cell will go away. Then you add again the substrate and do it again. Again. For 1 week, you can do the reactions inside the cells. And eventually what happens is that you have too many cells to do anything, and you don't have enough space. But for 1 week, you can have the palladium nanoreactors inside a cell working. Okay, so it's not something that works many times in a test tube, but it also works inside the living cells. So you see, this is the run one, then you let 6 hours, it gets clean, do another one, and so on. And then the editor of Nature Chemistry said, okay, I'm not going to publish this unless you do something with animals. This was after 1 year of revision. And then we say, okay, let's do something with spheroid, just to convince him. As you see, it was not published in Nature Chemistry, okay? But we did it with spheroids. And this is kind of spheroid. So we have a lot of palladium nanoreactors inside this spheroid. And you see them because of the fluorescent. We put a fluorescence tag in the MOF here. And in this spheroid, you see different cuts of the spheroid. When you go to the middle, you have less of these nanoreactors in the middle, okay? And then we did this also for, I think it was 10 days or something like that. Basically, you have the model of the spheroid, and then you add the product. It goes inside the spheroid. You produce the pink fluorescent. Then you let it rest for 1 day, and it is clean, self-cleaned at this point, and then you do it again. So even in a model like that, you can imagine that you could have even a kind of tissue-like model in your skin, and with this palladium reactor, you get injected that you only produce the drug where you have the palladium. Okay, it's not something that I want to have palladium inside my skin, but it works very nicely. And then to finish, I will talk a little bit about functionalization of MOF. That is something more recent, and we don't have all published. And basically here you have, this is UIO 67. It's a kind of zirconium MOF that we work with. And it's relatively easy to functionalize it with polymers, proteins, peptides, and anything. So zirconium MOF, they are very cool to functionalize. So the idea now is to go further and try to work on the surface functionalization of nanomorph as we used to do with nanoparticles, to direct them to the specific route, to target some receptors in some specific cells, and so on. Because MOF allows you to do all. So basically, you can put a lot of drugs inside, and then you can put, you have a very rich chemistry to work on the surface. So this is a work of Manuela again. Basically, I asked her to compare. So she did a research stay with David Feiden in Cambridge, and then she was working with this PCN222 and working in pegylation. And basically, what David Feiden developed is that phosphate interact a lot with the zirconium cluster that you have in this MOF. So it's a kind of click reaction that you can functionalize them with DNA, PEG ended in phosphates and so on. It's very straightforward with anything more or less. And I asked her to compare C-Fate, NU1000, PCN222, and UIO67, different chemistries, different systems in the nanoscale, to grow on top of it either PEG or this PMA. In this particular case, it was a polymer with a cycloctene group to do click chemistry on top of the MOF. So basically, once you have this cycloctene, I asked her to put some PEG, some mannose, and some cell-penetrating peptides. This is a long story, this cell-penetrating peptide, but it's supposed to be very efficient in bringing nanoparticles inside a cell and follow some routes that I don't have time to explain. So she did that. It worked very well. She has these nanoparticles, either with this octahedral structure like UIO, this rice-like structure like NU, or this PCN222, or this C-Fate. C-Fate is the one that we use from the beginning with the polymer, and then we do the encapsulation. When we do the encapsulation, we preserve the structure properties, optical properties, and so on, by X-ray, by BET, and so on. And then I ask her to compare internalization. So this is what we were doing 15 years back. So putting gold nanoparticles, same gold nanoparticle, put in different coatings, and see which one is better to bring cells inside, or to escape an endocytic route, or to do whatever. Okay. And with nanoparticles, we were making 2D libraries, 3D libraries by changing different aspects of hydrophobicity, elasticity, charge, anything that you can imagine. And in this particular case, we simply change the molecules that you have on the surface of the MOF to see if it has an impact on the amount of cells that you have inside. The idea was Z is SIF, U is UIO, because we wanted to compare similar shape and size. So this is without anything on the surface. And then when we put PEG, uptake goes down. Sorry. That is something that we were expecting. Okay, so we have the MOF, and this MOF has a complex chemistry on top of it, and normally they are positively charged. And then when you put the PEG, internalization goes down. And then we put mannose, carbohydrate, expecting that it will bring a lot of particles inside because these cells express a mannose receptor, but it didn't work. Okay, I don't know why. It's something we have to work on, because maybe the mannose goes is a little bit inside the MOF, or it's not outside, or I don't know, or we don't have enough density, or whatever. It doesn't work. That was what we wanted. But the cell-penetrating peptide, it works very well. So you have tons of particles inside. Okay. It's not about charge, it's about interacting with specific receptors and so on. So this cell-penetrating peptide is, so I have to shut up already. So basically we can get a lot of these particles. So the next thing is, and with this I finish, is to use membranes of one cell. So we have any type of cell, cancer cells, macrophages, or whatever. We remove the membranes, and then we make nanoparticles. So now we want, instead of put mano cell penetrating nucleic chemistry, we want to coat the MOF with these cell membranes, because there is something that is very interesting for me, that is something called homotypic targeting, that basically, if you make nanoparticles of one specific cell type, if you work with cancer cell lines, you make it from HeLa, or other one. And then you add it to different cancer cells, it always goes to the cells from they were originated. Okay, it's something called homotypic targeting. So basically, they don't want to go to macrophages, they want to go to these specific cells. It's something called homotypic targeting. So in this particular case, we wanted to coat our MOF with this kind of cells, and something else to make it fusogenic, because we didn't them to end up in endocytic lysosomes. It's another paper. This from, because we make this biomimetic cell surface that fuse with the cell membranes, and then the content is inside the cytosol and not in endosomes or lysosomes. It worked very well. I have data with a amount of zirconium inside the cells, but basically this is the easiest thing. You have the MOF with something that is pink, Crecil violet, and the coating with the cell membranes. And this is what you get when you don't make it fusogenic. And when you make it fusogenic, you see that the content is spread in the whole cell. Okay? So it's basically because it didn't follow a endolysosomal route. You can also see this with TM. This is when you have the coating that is not fusogenic, and you see the MOF inside vesicles. And when you have the fusosome, you don't see it in vesicles anymore. Okay? And then we loaded a drug with this particular system. With the fusogenic ones, we completely destroy the tumors, and you can follow this over time. So basically, these are another example of functionalization. In this particular case, is a biomimetic functionalization of MOF or nanoparticles or anything that you want to do. Okay, it's using cells to do this coating. And with that, I'm done. I want to thank the collaborators, Patricia, José Luis, Sara, and so on, the people of the group, and thank you very much for listening to me.
Thank you, Pablo. Before starting with questions, I just want to know if anybody is interested in talking to him later, come to the front after we finish. So now it's time for questions.
Just a curiosity, I really enjoyed the talk, really interesting. And you were commenting that if you want to activate these systems, you can use either radiation like light or other kind of stimuli. Have you tried like a magnetic field, for instance?
Yes.
Like, and how does it work, if may I ask?
So basically, we need nanoparticles that absorb a lot of alternating magnetic fields.
Yes.
These alternating magnetic fields, they get absorbed inside the nanoparticle, and then it releases the absorbed energy in the form of heat.
Yes.
And this is what we use, as we do with light, with alternating magnetic fields. The other one that you have to time to go through is not absorbing and producing heat, is rotating magnetic fields that they go at very low frequency, and they are able to, the particle is able to rotate with this. I don't understand very well how this works, but it also works.
And for instance, when you apply the magnetic field, like you localize like the heat in the nanoparticle region, that you don't damage the surrounding
you have
scaffold, like
you have to adjust it.
Okay.
It's,
so you can
it's like with light.
Okay,
I mean, if I put light, I can bubble and boil the cell media.
So you graduate.
You graduate it just to have the effect that you want, drug release or. But magnetic nanoparticles, they are typically used for magnetic hyperthermia, and what you want is to destroy the.
Yeah.
Yeah.
Thank you so much.
Hi, thank you very much for the talk. Very inspiring. I was curious about the synthetic route that you've used for the MOFs, that you start from the seeding, no, the nanoparticle, maybe gold or any other metal. So have you tried to do it in the other way, but functionalizing the MOF with inorganic nanoparticles around, so they have kind of interactions between them.
Yes.
I'm explaining.
Yes. The thing is that nanoparticles cannot go inside the MOF, because to have a nanoparticle, you need more than 2 nanometers. Well, I mean, you can have clusters as well, of course. But you can decorate, for instance, the surface of the particles.
So they do not collapse the MOF,
No,
right?
No. I didn't mention, but a MOF is extremely robust. In general, they sustain 500 degrees, 400 degrees. They are crystals. They are very strong. You can do whatever. I mean, Pepe's MOF, you cannot destroy them even with fluoric acid. Is terrible. Yeah.
Hi, thanks. Really nice presentation and very good results. I have a few questions, but maybe a couple. On your palladium catalyst, you say I wash and I take it out. What is that you take out?
So basically, we do. I explain it very fast. So basically, you have a cell culture, either a spheroid or whatever you want. You feed the MOF. You let them for 6 hours, 3 hours.
Yeah.
Yeah. You have them inside. Then you wash everything that is outside, and then I get the product from José Luis. That is this cage fluorophores. I add it. I let it for 1 hour, 2 hours, 6 hours. It depends. If you leave it, it's a concentration dependent thing as well. Yeah. Okay. So you let it, you remove it, and then you see how much of the product is inside the cell.
Exactly.
Then at this point, I don't have substrate outside. I let it in the incubator again, and I observe it 6 hours later. And 6 hours later, the product that is inside is away.
So, but the product that is away are the MOFs?
No. What I produce, the fluorophore. Okay? So I didn't understand this at the beginning.
Okay.
I say, why?
Good that I am not the only one.
Because I never work with, because I don't work with small things. But then it's something that the organic chemist, José Luis told me, yes, you can do a reaction, but if you remove, because you have different concentration of the product outside the cell and inside the cell, it will go away. And
but you telling what you're telling me is that your cell have uptaken a certain amount of MOFs, and then the cell is expulsing them?
Not the MOF.
Only the palladium.
Because I have a substrate. Imagine it's like you have an insert here.
That was the question. The MOF remains inside.
Yes, because I can work and do the same reaction for 1 week.
Okay.
I mean, I was expecting not that it works so well because cells divide, but it was working amazingly.
Okay. Amazingly. Yeah. I think now I get it. And the ones that they have gold inside, your gold particles are rather big because you want them in the infrared, so they are stars. So are you worried by the amount of gold that stays in the cell?
Yeah, I mean, absolutely. This is a proof of concept just to demonstrate that I'm able to do that.
Okay.
Some other systems, they use ultra thin rods of 2 nanometers with 20 nanometers in long that you also have absorption in the infrared and so on. But this work, I mean, I don't see this doing any therapy in the future. Just to demonstrate that I'm able to do, I didn't have time to, but what I was saying that I'm able to produce a thermal gradient at the nanoscale, nobody believes it. Okay, if you talk to a physicist, he will tell me, no way. Heat transfer doesn't work like that. But this is a long story. The reason by which you can create a thermal gradient or whatever I'm creating to expel something that I encapsulated in a porous material.
Thank you.
Also, the temperature doesn't exist at the nanoscale, strictly speaking. So.
Any other question? I had just one comment or curiosity. When you say that the CF8 is not stable with CO2 in the presence of water, right?
In any.
With gas? No, but I mean, if you avoid the humidity, because the CO2 will go to coordinate, maybe it's CO2 plus water makes some base like bicarbonate, and that will be the base that will remove the.
Yeah, probably is this.
Probably, no?
Yes.
Yeah.
It's carbonate.
Yeah. If no more questions, let's thank again Pablo for his talk.